All right, class, we're going to get into the urinary system. Every day, kidneys are going to filter gallons of fluids from the bloodstream, taking waste and excess ions that leave the body by the urine while returning needed substances back into the blood. The best way to explain or understand conceptually what the urinary system is that it is like a water treatment center or facility. Think about it. You may not know this, or you may not even want to know this, but the water you're drinking started out as the water in your toilet. So what I mean by that is, yeah, it's the same water, but it's water that contains urine and fecal material. And that dirty water, if you will, is sent to a treatment facility. It is cleaned, it is treated, it is processed in any manner and comes back and you use it. In that mechanism, in that thought process, that is exactly what happens with the urinary system and blood. Okay, that's the best analogy. So let's talk about what's included in the urinary system. Now I know in lab you talked about all these things and you looked at a kidney close up. So it includes the kidneys, ureters, urinary bladder, and urethra. They're all making up that urinary system, either as a storage reserve for urine or transportation channels to carry the urine. So let's talk about the functions of the kidney. So kidneys are going to have several functions. The first function is to filter plasma as it becomes filtrate. So you'll see that within the kidney itself, the smallest component that makes urine is called a nephron, and we'll talk about it in a little bit. So the nephron is going to actually filter blood plasma, and it's going to separate waste and making it leave with the urine, and then good solutes, things your body may need, puts back in that bloodstream, because those blood vessels are right there inside the kidney to do its work. Okay. Some of the waste products we'll talk about are called nitrogenous waste. And again, we'll talk about that shortly. And just remember that if there is a buildup of these waste products, then it is going to be a serious situation, because you can potentially die from it. Okay. The second function is to regulate blood volume and osmolarity. Okay, osmolarity is the number or amount of solute particles present in 1 liter of solution. And you can usually judge that. If you look at urine, technically the darker it is, the more solutes are present, Okay. The third function is to produce hormones. These hormones regulate the process of urine filtration. And we'll talk about the hormones again in a little bit. Fourth function is to regulate acid-base balance of the body fluids. So your body, let's say if it's too acidic, you already know that blood is going to be filtered in the kidneys. You know that you have blood circulation into the kidneys. Okay, so your blood, your kidneys, the blood entering the kidneys allows for the kidneys to identify and recognize that there is a component of the pH being off. And in this instant, the kidney itself can release buffering agents to therefore balance the ion concentration. Fifth function is to detoxify superoxides, free radicals, and also drugs. So basically what it does is it brings, so the urinary system, the nephron itself, brings in these components that are dangerous to the body. It binds with them and it releases them into your urine. So superoxides, you know, oxygen is written by O2. Superoxides would be like O4, O6. Also, if we're talking about free radicals, those are ions that are not bound to anything, and both can be very problematic. Okay? And of course, drugs. That's one way to kind of get rid of drugs. Think about it in this manner. You know, why are drug tests done with urine? Well, there's your reason why, because it's drugs that are released out in the urine. Okay? So as far as kidneys function and structure, kidneys are bean-shaped organs. They lie against the dorsal body wall. Right is slightly lower than the left because of the liver. It's found in the superior lumbar region around the 12th rib. The size of it, which you can see in that top right-hand image, the size of it is about the size of a bar of soap. Medical identification, or the medical, excuse me, sorry, misspoke. The medical identification called the renal hilum, which is, you can see in that bottom picture, that space. You can also see that hilum is represented in that left image. You can see where the blood vessels are entering and exiting. You can also see where the ureter is exiting the kidney. So the hilum is very important because it represents the area where things get into and out of the kidney itself. Okay. Also, nerves will enter there. Okay. So that hilum is really important. So if we were to cut open the kidney lengthwise, which you can see in both pictures below, the outer layer, you can see that basically the tissue has two different colors. And you saw it on the models as well. Those pyramid-like shapes, that is darker in color, and it is painfully obvious that even with a human kidney dissected, you can see there is a difference in color pattern. So those triangles are made up of tissue called medulla. And then the tissue surrounding it is significantly lighter in color. That's called the cortex tissue. So we have two tissue types inside the kidney, medulla and cortex. Okay, so also those pyramid-like shaped, made up of medulla tissue, they're called renal pyramids. Pyramid is triangle in shape. So if I were to ask you, or if in lab they were to ask you, what is this called, you would say renal pyramid. If they say, what tissue is it made up of, then the answer is medulla. Again, we're talking about these things. Shoot. All right, so I switched it. So I'm going to, by accident, switched it. So I'm going to keep talking about the different structures, or I'd have to redo the whole PowerPoint. So we've got those two shapes. We've got the two different types of tissue. Okay. Those pyramids, those renal pyramids, they're actually cup shaped. If you were to look at them or think about them, they're the shape of like a petal of a rose. And it comes to that point. Well, there's ridges, little tiny natural ridges within that pyramid shape. Those are called papilla. So what happens, and you'll learn urine production obviously, but what happens is urine is made, the tiny droplets, the minute droplets from the urine being made slides down or rolls down those papilla, and then it's caught at the very bottom of the pyramid where it has that pointed shape. And then once all the droplets collect to a certain amount, then they'll go into the sinus, the renal sinus, which are those cavities, those tunnel-like things, and then collects and then goes down the ureter. Okay? All right. So apologize for that. Again, what I'll do is I'll upload an image I'll identify all the structures so you'll actually be able to see and compare contrast what I've talked about. So we're into this slide for the kidneys coverings. Obviously, they're essential for survival. So the kidneys themselves, if they're not functioning, you already know that you go into kidney failure, and then your blood isn't being filtered and you have nitrogenous waste products just collecting in your body, which is extremely dangerous. Okay, so we have to protect them. They have to have several things that will protect them and keep them working correctly, and protect them obviously from danger or damage. So if we talk about it, the coverings, the first thing is the capsule. So the outer layer of the kidney is made up of a capsule. The capsule itself is really difficult to cut through. It feels like a hard rubber ball. And that hard rubber ball, that covering will protect it from injury. That's one way. That's still not enough. Surrounding each of your kidneys is going to be an obscene amount of fat. So that fat is an extra layer of protection. Still not enough. We want to keep them in a localized position. We don't want them to move. We don't want them to be affected by friction, if you will. So it holds it in place is called renal fascia. And you have fascia a lot of places. If you've ever kind of, cooked some meat and you kind of notice that iridescent, slightly transparent tissue, if you will, you try to pull the steak away from the bone, or even chicken from the bone, and you see this iridescent, that's fascia. Well, you can see it on this image below, holding that kidney in place. That is the renal fascia. So those three components are protective measures to kind of protect the kidney from damage. So I like to give diseases and disorders because I think it's important to try to understand what could happen if it goes wrong or if there's problems. So this first disease or disorder is hydronephrosis. Hydro refers to water. Nephrosis, remember I told you the primary urine producing structure in the kidneys is called a nephron. So hydronephrosis means literally water in the kidney. Okay, it's distension and dilation of the renal pelvis, which is that center portion of the kidney. So it's now collecting water and storing water when it shouldn't be, and it kind of blows up like a balloon. And in that bottom picture, you can clearly see the right one is a kidney, a true kidney that's just been dissected open. And then the other kidney does have the hydronephrosis, and you can see it looks like a giant water balloon. And this basically occurs because there is some sort of occlusion. There is some sort of blockage that doesn't allow urine to leave the kidney. I mean, signs and symptoms depends on whether it's acute, meaning it just started, or you've had it for a long period of time. I mean, pain, nausea, vomiting, UTI, stones, fever, blood and pus, and what urine you can produce. Prognosis depends on the removal of the obstruction and drainage. So literally, we have to figure out what's causing it. So somehow we have to, once we figure it out, we have to make the occlusion or the blockage be gone. If we can't get it to work, we're going to have to remove a kidney. Now, of course, you can survive on one kidney. It just takes longer to process the blood, but you still can survive with it. There is that image of the hilum, which you can see renal artery and renal vein. We just happen to be on the left kidney, but you have a left renal artery, left renal vein, and then on the other kidney, you have a right renal artery and a right renal vein. About a quarter of your blood supply within the body passes through the kidney each minute. So therefore, it's a quarter blood supply. That means that within 4 minutes time, you'll have all of your blood, which is passed through that kidney. That's amazing. So at that hilum area, the arteries and veins separate, they branch off, and you can see they go around each of the renal pyramids. And the space between each renal pyramid is called a column. And you can see the blood vessels there. And shortly, when we talk about the nephron itself, you'll see that is where it gets into the nephron. It's filtered and allows for urine production. So the nephron, okay, is important for urine production. In fact, its primary function is urine production. And each kidney contains about 1.2 million nephrons. They are the structural and functional units of the kidney. They're responsible for making urine. They consist of blood vessels, both afferent and efferent. Remember, afferent goes towards it, efferent goes away from the nephron. There is a glomerulus, which is the filtering mechanism for the nephron. And then you have renal tubules. You have a proximal and distal convoluted tubules, which we'll talk about in a second when I get to the position where I can show you the kidney map. So, okay, in the glomerulus, you have that twisted knot of capillaries, which you can see on the right-hand side. Okay, the glomerulus, okay, has some protective boundary, if you will. It's a protective membrane. It's called Bowman's capsule. So that whole structure itself, if I were to ask you what the whole structure itself, it's called the glomerulus. If I ask you what the protective coating is on the glomerulus, your answer is going to be Bowman's capsule. Bowman's capsule main function is to filter, so there must be space to get through. Think of it like a screen door, that screen material, or a colander, the thing that you strain spaghetti when you're cooking spaghetti. So it has to have these holes in it to allow certain things to exit through it. But this is done because the Bowman's capsule is actually made up of living cells. The living cells are called podocytes. And if you will, imagine a podocyte to look like an octopus. So what happens is these octopus-like structures sit together one another, and the tentacles, if you will, and they're not tentacles, but the tentacles, if you will, line up in a fashion so it looks like a mesh net. There's spacing between it. It's not lined up perfect at all. Okay? They're intertwined, if you will. Okay? And this is what's going to allow for filtration, because only certain things can get through. Okay? So if we look at a true kidney map, with the kidney map, okay, so on that left-hand side of the PowerPoint. Okay, so you're looking at the structures in that first upper left-hand diagram. You're looking at the structures that make up the urinary system, and you can see that the kidneys are what we're going to focus on. So in the next one next to it, you can see that the kidney dissected. So you can see the renal pyramids made up of medulla tissue. You can see the renal cortex, which surrounds the medulla. And if we blow that up even farther, so we're looking at that triangle that we've kind of pulled out, that sectioned out. So then we're looking at that at a closer level. Okay, so we're looking at it, and you can see these twisty things that are found in both the cortex and slightly in the medulla. So if we blow that up further, which is the same picture that I've blown up on the right-hand side, there is the kidney map. You can see the glomerulus, and you can clearly see that blood vessels are going into it. So you've got the glomerulus, and see that twisting turn of material. Initially, that first bit of twisty turn tubules is called the proximal tubule. Well, it's proximal to it. That means it's close to the glomerulus. So that's the proximal tubule. The proximal tubule then dips down into this loop. Well, that loop is called the loop of Henle. So we got the proximal tubule that goes in that one direction towards the loop of Henle, and then it comes back up into another twisty type tubule. That's further away from the glomerulus, and therefore that twisty tube at the end is going to be called the distal tubule or distal convoluted tubule. And then by that point, we're now going to dump whatever has made it all the way into a collecting duct. And again, we're about to talk about how urine is made. But there is the kidney map. Now you need to understand what it looks like for not only lab, because you could be questioned on that, but you also need to know about it when we're talking about the lecture component. So it's real important for both. So let's talk about urine formation. Urine formation itself requires 3 steps, and it benefits both in a way that allows for things into and out of it that are important and necessary. It allows for things that shouldn't go out of it to get out of it. So you're going to see quickly how important is that that urine formation taking a 3-step process. The first step is glomerular filtration. The second step is tubular reabsorption. The third and final step is going to be tubular secretion. All right, so let's actually get into the physical steps. So the first step is glomerular filtration. I can tell you right now, step 1 takes a couple slides to explain. It seems to be, how should you say, the most difficult as far as the process, and we'll just leave it at that. So the glomerulus acts as a filter. It's non-selective, which means what's going to be processed through it isn't specific. It's non-selective. So it's non-selective. It's a passive process in which fluid passes from the blood into Bowman's capsule. And in Bowman's capsule, there is this 3-layered or 3-barrier system that allows for specific particles to get through each layer. And the best way to kind of think about it is if you were, let's say, looking for gold in a creek. So the first screen might have big holes. So you've got the bigger stuff. And then you want to put it through something smaller to get the details. So the next screen you dump it into has holes that are a little bit smaller. And then finally, if you're really looking for teeny things, you're going to actually dump it into a very, very small hold screen. So if you notice, each screen I went through has smaller holes, if you will, to pass something through. So the idea of this 3-barrier system, each layer that something can get through, the holes or pores get smaller. So only certain things should make it into the second and third process of urine. So the 3 layers, which I've put a kind of picture there with identification of them. The first one is the foot process. Again, remember I said the outer cells that make it up are alive, and they're called podocytes. And remember I said they have like tentacle-like structures. Well, you can see in the foot process picture and what the arrow is pointing to, they look like tentacle-like structures. So any holes between that twisting, if you will, of the podocytes is the first layer. If it makes it through the first layer, then the next layer is fenestrated epithelium. Fenestrated is a fancy word for holes. So if it makes it through the foot process, the next layer is going to be the fenestrated epithelium, which the pores or the holes themselves are smaller. And then finally, if it makes it through those 2 layers, the last layer that it must get through is called the basement membrane. And like I said, the basement membrane, which is represented by that pale pink color, is going to have the smallest pores for it to pass through. The idea is passing through things that should go into the urine rather than things that need to stay into your bloodstream. Okay, once the fluid from the plasma, once whatever can pass through those 3 layers continues on. Well, that fluid that has made it through those 3 layers is called filtrate. Think about it, it's been filtered. So the fluid that makes it through those 3 layers is now called filtrate. Okay, so basically blood plasma without blood protein and blood cells, which are too large to pass the barriers, so they're forced out of the blood through the capillary walls back into the renal tubule. So the idea is only small things should make it into the filtrate. And we're talking about things like water, electrolytes, glucose, amino acids, lipids, vitamins, and nitrogenous waste. Ideally, that's all that should be entering the filtrate, or making it to the filtrate. However, there are sometimes things that make it through all these layers, which they shouldn't, like protein or blood, because they're technically too big to pass through. Finding them in a urinalysis, that would explain where there is a problem. Something has occurred. That's why a urinalysis is quite effective. Okay? Think about it. A UTI is a urinary tract infection. And if that UTI has climbed all the way to the kidneys, meaning the bacteria has colonized and moved up the ureters and colonized again inside the kidney where you truly have a kidney infection. And I know some people use that interchangeably, but a kidney infection is much more severe with a very high fever. You'll notice blood in your urine. This is something where you need to actually go to a doctor and be physically treated. It's not like a UTI where you just get some antibiotics. This is much more significant and more problematic. So what causes, or one of the main reasons that would cause such a problem as blood or protein making it into the urine leads to suggest that there is some sort of kidney infection in that area. So the infection has caused the spacing of pores to be larger, therefore allowing things to get out into your urine that shouldn't be. It also can be simply from damage. Some sort of trauma can cause this to occur as well. And then serious issues like, diabetes, overindulging or overeating of proteins. These are all things that seriously can cause problems. So there has to be some way to allow this naturally to be passive. And you always know that things that move for a specific reason. So generally with pressure, it's a pressure gradient. Things move from a higher concentration to a lower concentration, which is a passive process in which normally that's how plasma moves into that glomerulus. So that passive process allows it to occur. And there needs to be this push, this movement to occur, and that's that filtration pressure. It's not active, so therefore it's not requiring energy, and it still is truly passive process from higher to lower concentrations. This pressure allows for things to move with the barriers. So outward pressure causes the main force pushing waters and solutes out of blood into the filtrate. This occurs on the entire capillary bed. The inward pressure, there's two inward forces which inhibit filtration or filtrate formation by opposing outward pressure. Normally around 10 micrograms determines the glomerular filtration rate. So there has to be, and you can see in that image towards the bottom, there has to be a balance. So you've got one inward pressure that inhibits filtrate formation, and then you have two outward that counterbalance, allowing for filtrate formation. That GFR, or glomerular filtration rate, is the amount of filtrate formed per 1 minute by both kidneys together. So it's a measurable amount of filtrate, and the idea is if we were to actually measure it, it would give us an idea of the functionality, if you will. How well is your kidney doing its job? GFR is roughly, and we're talking about, I don't want to say a male, but the average individual, average healthy individual, is about 125 milliliters per minute. Therefore, that means about 180 liters per day. That's how much filtrate you are making per day. I hope you're not urinating 180 liters per day. That's not normal. So almost all filtrate is reabsorbed, allowing for only 1 to 2 liters of urine per day. So that's what the normal natural amounts is, somewhere in that range between 1 and 2 liters. So it is a measurable amount. There is a way to see, okay, you're having issues. Well, what is it? Is there an infection? Is there a blockage? If we go through all the different portions of the urinary system. If it's not that, okay, what is going on with the urine production? Is it one area or one kidney that's causing the problem, or is it both kidneys? And that's a way we can kind of determine, is it one or the other, or is it both causing this kidney failure and issues? And that, like I said, that measurable amount is a GFR, glomerular filtration rate. Okay. So the nephron has to have several ways to kind of adjust on issues. Okay, so there has to be several ways to kind of regulate what's going on, and there's regulation by the nephron itself. There's renal or kidney ways to actually regulate it. So the first way has the nephron actually preventing drastic changes or very aggressive or very fast changes in that glomerular filtration rate. And this occurs when blood pressure rises. Think about it. If blood pressure is going up, then that GFR is going to have more pressure. So it's going to affect urine production. So with the nephron doing it, it needs to protect and keep the GFR in a constant rate. It can't allow fluctuation. That's dangerous. So what happens, or what can it do? Well, if it constricts the afferent arteriole, think about afferent ascending into the kidneys. Afferent, so this is the arteriole that's entering in the kidneys for filtration. Okay, so if we constrict it, we would therefore lessen the amount of blood entering the kidney. And again, for constriction measures, you know there has to be a small or thin muscle surrounding each of these blood vessels. So the idea is, for constricting that arteriole, that means we're lessening the amount of blood entering the kidney. Well, that helps regulate the GFR. That'll actually lower it. Another way is to dilate the efferent arteriole. So we're constricting the afferent arteriole, which means less amount of blood is getting in. Well, we need whatever's already in there to get out. So what do? We dilate the efferent arteriole. We allow more blood out. That's how the nephron itself gets involved with the GFR. There is renal autoregulation. There is a way that it naturally can a constant GFR. So we have the ways that the nephron does, and then the renal kidney itself has the myogenic mechanism. Those are that is where smooth muscles constrict and relax, controlling blood flow into the glomerulus. Okay, so that means it constricts, relaxes, constricts, lowers it, the blood flow in, relaxes, increases the blood flow in. The second one is tubular glomerular feedback mechanism. That's autoregulation by flow. So if GFR is increasing, not enough time for reabsorption, and the concentration of sodium chloride or NaCl in the filtrate has to remain high. Then these specialized cells called macula densa release constrictor chemicals, causing the arterioles to constrict, therefore decreasing the flow into the glomerulus. Right? So that GFR, we already know the nephron is involved with controlling it. Not enough. We also know the kidney itself is involved with controlling it. Again, that's not enough. So there also is an extrinsic control. There's control outside of the kidney itself. And that's where we have neural regulation. That's where the brain comes in to play with it. Okay? The sympathetic nervous system gets involved and causes the release of hormones. So there is that identifying receptors that realize what's in the blood and what is not in the blood and what should be being kept in the blood and released in the urine. So there is this regulation. So I need you to think about what type of regulation involves the nervous system. So there's two things. That it's not neural regulation because, you know, it's not necessary unless you're feeling bad. Let me rephrase that because I'm phrasing it wrong because I want to say 30 things at once. You know how I am. So there's two mechanisms that allow for this control, this extrinsic control. So if it's neuroregulation, what I mean by neuroregulation is that it's the sympathetic nerve fibers. You feel bad. You know, when you're hungry and you haven't eaten, you feel terrible. Or when you eat, you feel bad. Or if you have blood sugar issues, if they're high or low, well, there has to be neural control. There also is humoral control. Remember, the humoral control is looking for ion fluctuations. Well, those two things cause negative feedback to occur until it's fixed. So again, we're talking about that feedback system in both instances for survival. The hormone specifically we're talking about is aldosterone, angiotensin, and then renin. Okay? Okay, so just to reiterate some of the things that are really important, remember that we're talking about GFR. So remember that when we talked about GFR, you don't want fluctuation. So if GFR is high, then urine output increases. And therefore dehydration, electrolytes are lost. And that makes sense. It's making more filtrate than necessary. You're going to pee more, and it's going to cause an imbalance in the blood volume. If GFR is too low, then wastes are not being excreted out. So you build up waste products into your blood, causing some significant issues with that. Okay? So the second step, so that's all involved with the first one, glomerular filtration. So that was the first step. Now anything that makes it through all that and is now filtrate is going to go into the tubular reabsorption. Tubular reabsorption is what leaves as urine as the substances you don't want, waste products. But body needs some things in the filtrate like water, glucose, amino acids. So what happens is this filtrate now makes it into the proximal convoluted tubules. But what happens if you suddenly can't eat? You're in a desert and there's no water. So we don't want you to urine more. We actually want you to urine less. Your body wants you to urine less. That way we can preserve blood volume. So what happens is the water that you need is pulled out of the urine in this second step and put back into the bloodstream. That's tubular reabsorption. Things that you need that you don't want it to go out in urine is pulled from the proximal tubule and put into the bloodstream. Man, that's a great backup system if you think about it, because it ensures that things that'll maintain survival get back. Okay. This is going to be done by different mechanisms. So solvent drag is the first one. Okay, that is driven by osmotic pressure in the capule or the capillaries surrounding it. A solvent drag is when water is reabsorbed by osmosis, and then it carries all other solutes along. The second one is active transport by sodium. That involves the sodium pump. What happens is through these membranes, sodium is going to be pulled out of cells against concentration gradient, and this does require ATP, so it is an active transport. So it's active transport. And remember, wherever sodium goes, water is going to follow. Okay. The third one is secondary active transport of glucose, amino acids, and other nutrients that your body will need. There are various other co-transporters, if you will, that carry both sodium and other solutes. So up until now, the last one that we talked about, number 2, that was only working on sodium, and that was using the sodium pump. So this other one is going to have different transporters that not only regulate the sodium, but are going to regulate other nutrients or ions that your body requires. Okay? The fourth one is secondary water reabsorption by osmosis. So you had that one that was osmosis, and that was a regulator of it. The next one is just in case the backup system, which is this reabsorption making both intracellular and extracellular fluid hypertonic in the tubular fluid. Therefore, water follows sodium into the peritubular capillaries. So basically what happens is to move the water, we have to make them hypertonic. So you need some sort of salt. When we make it hypertonic with the salt or sodium, then what happens is where it's going, water is going to follow. So we're going to store the water in the intracellular fluid of the cells itself, and we're also going to store the fluid or water in the fluid that surrounds cells, which is the extracellular fluid. The sixth one is endocytosis of the larger solutes that can't move in this process. So endocytosis, remember, is the bringing in, the surrounding and pulling it in. So remember, the glomerulus is going to filter a small amount of protein from the blood. And then it is moved by endocytosis, which hydrolyzes it into amino acids. So it changes it it breaks it down to amino acids. And then it is released into the extracellular fluid by facilitated diffusion. Again, so we're moving everything from one to another, allowing for it to survive. That is tubular reabsorption. So yeah, look at these different things. And most of them are self-explanatory, but that makes sense in these proximal tubules. There is that safety net where we don't want to pee out things if there's a sudden emergency. We want to be able to put it back into the blood volume for use for our body for survival. Okay, so that's why we have that reabsorption. It can be put back in the body. But what happens if everything makes it, that filtrate makes it through the glomerulus, makes it through the proximal tubule? That's what's left. Well, what happens if there's an emergency that we need to put it out of your body? We need to get it out. That's where step 3 comes along. Step 3 is going to be this tubular secretion, and it's going to happen in the loop of Henle. Okay, so step 3 is the last step. It is the opposite of step 2, or tubular reabsorption. It does occur in this loop of Henle, and the idea is this is a place where your blood can directly dump something that you don't want to have in your body into the urine. So step 3 really bypasses the filtration of step 1, the glomerular filtration. So this bypasses this, and it's like an emergency measure where it can be stuck in your urine and released. And this could be protein. You ate like 50 grams of straight protein and nothing else. Well, that's too much protein for your body. It's dangerous. Let me stick it in step 3. I've taken drugs. It can hurt your body. Well, I don't want it to go through that entire process of trying to make it through into the filtrate by steps 1 and 2. I need to get it out in your urine and preserve your body, protect your body. Well, it's dumped immediately into step 3, into that loop of Henle to be fixed or pushed out in your urine. Brilliant system. Filtering naturally with step 1. Pulling things out in a situation where we need to keep your internal environment, blood volume at normal levels. So this can ensure that you're not going to lose blood volume because of urination. And then step 3, dear God, what if you ingested too much of a drug? And I'm not saying it's going to save you, but there has to be a way to get rid of the drugs out of your body and hopefully decrease their side effects or the problems that can occur because of you taking that drug. I mean, it's brilliant. So I've mentioned nitrogenous waste a few times, and there really is 3 nitrogenous waste products. Okay, so 3 nitrogenous waste products. Okay, the first one is urea. That's formed by the liver as end products of protein. It breaks down amino acids, and it's used as energy. Okay, the second one is uric acid. Uric acid is released when nucleic acids are metabolized. Okay, the fourth one, or the third one is creatinine. Okay, creatinine is when creatine is metabolized in muscle tissue, this is released. So all of these things, there's normal levels that are measurable in your body. High levels of these explain that there is some problems going on. This is aids in diagnosis of issues. So you've heard of diuretics. In fact, I mentioned it when we talked about the endocrine system. Those are chemicals that enhance urinary output. And most people over like the age of 60, or, it could be earlier, but let's just say 60, have to take diuretics, because just like anything else in your body, kidneys are going to age too, and their effectiveness decreases with age. Doesn't mean you're going to die or have kidney problems, but they just don't function quite the same. So you're given a diuretic to kind of enhance urine production, so you are urinating at the levels that you were urinating when you were younger. The idea or how this works is one of two ways. There are diuretics which inhibit sodium reabsorption. These are the diuretics that you can purchase at Walmart. So remember, where sodium goes, water is going to follow. So if sodium is put into the bloodstream, or sodium is put out of the bloodstream and forced into the nephron, then water is going to follow, and therefore your urine output increases. That makes sense. That's brilliant. That's something you can get, at Walmart, and that's the way it's done. And that's sometimes taken by females with menstrual cycles where they're retaining a lot of salt and kind of get bloated during that menstrual cycle. And some people will take that diuretic, which alleviates or causes more frequent urination. Would I recommend you staying on that diuretic to try to allow for you to be less weight, so you don't have any much bloating? No, that's actually dangerous for long-term use, because you're going to get dehydrated. The other means of diuretics, this would be in the manner of a prescription by a doctor, and that's going to inhibit the release of antidiuretic hormone. Because remember, antidiuretic, that means that urine is going to decrease. It's going to not have the high amounts. So if we stop antidiuretic hormone from being released, you're going to urinate more. Okay, so that's the two means of diuretics. Okay, there's something called renal clearance. Okay, I know you love math, so of course I had to include this for understanding. So renal clearance is the volume of plasma from which the kidneys completely remove, the volume of plasma from which the kidneys completely remove a particular substance in a given time. So we're measuring how much something, like we can say salt, how much salt, if you will, and that's only one example, is removed from plasma in a specific time frame. And there's the formula for it. So C is going to represent renal clearance. So renal clearance equals U is the concentration of a particular substance in the urine. Urine. So it refers to the concentration of a substance in urine. It can be anything. It can be amino acids. It can be protein. It can be salt. It can be potassium. So it represents a particular thing, the thing we want to measure, that solute we want to measure. So that's the concentration of that particular thing, if you will, we want to measure in urine. So we multiply that concentration of substance in urine by V. V represents the flow rate of urine formation. How fast is it moving? How much is being moved for urine formation? And then P, we're going to divide that number. So U times V, so the concentration of a substance times the flow rate gives you a number. Well, then we're going to divide it by P. P is the concentration of that same substance found in U in blood plasma. So you notice how it's almost like we're negating or canceling it out. So the substance is measured in urine, and that same substance is also measured in plasma. Do you see? If it's equal, then all we know is the flow rate. So if U and P is the same number, well, if U is divided by P and it's let's say 100 of U and 100 of P, that equals 1. So then the answer would just be the normal flow rate. So you can see if it varies in either of the two, it's going to give you a different number. That's what we're looking at. So for example, if we talk about just, I'm picking simple numbers for understanding, U is going to equal 5. And then I'm going to put a little X for multiplying. Let's say that V equals 20. Right? I can't write, I'm sorry. So therefore, U times V equals 100, right? And let's say that P is 50. Then your C would equal 2. So then your renal clearance is 2. Okay? So it's not a difficult formula. Just make sure you understand what each thing means. Okay, so urine. I know you didn't get the opportunity of doing a urinalysis because of the online class, but you were able to kind of see what it was when I demonstrated a urinalysis, how it is. And you know, urine itself generally ranges from a clear pale color, so a light yellow, to a deep yellow. That's normal and natural. And remember, the color is dictated by solutes present. And also another thing that dictates the color, so the deeper yellow color, the really school bus yellow color, is going to be dictated by the destruction of hemoglobin. So red blood cells contain a protein called hemoglobin. When the red blood cell dies, we have to get rid of that hemoglobin. We have to break it down, and it produces urochrome. urochrome, urochrome. So that urochrome is also what gives it that color. Okay, it's also affected the color by foods you eat or drugs you're taking. And I'm not saying illicit drugs. It can be just simply medicine that you've been put on in a prescribed manner. So like foods, for instance, asparagus. If you eat lots of asparagus, there is going to be a greenish hue to your urine, and it's going to smell bad. So like in that urinalysis, we talked about or I talked about in the video, that we can detect certain things being present in a urinalysis. With that dipstick that I used, with those colored squares, we can see precisely how much of drugs are present, how much of ions are present based on that. And we also could say that just looking at the color dictates one thing. Smelling your urine, which, you know, I don't imagine you do frequently, but if you smell your urine, you can see that that can be an indicator of problems. Like if there is a nasty aroma, and I hate to say this because you're going to go ew, but like a fishy smell. So a fishy smell can be one of two things. It can be an STD, or it's also an indicator that you have an infection going on. And then if it smells sweet, that's a real good indicator that there is too much glucose present. You're in ketosis. So again, that's really important. The urine itself is full of water and solutes. Urine weighs more if it is more dense with solutes. So if we're looking at pale yellow urine, if we're talking about deep yellow urine, there is a varying weight, if you will, towards it. So the weight is called specific gravity. Specific gravity of water is 1.0. Specific gravity of urine ranges depending on how much solutes are present. 1.001 to 1.035. That's the normal range of specific gravity of urine. So it's a good measuring. Urinalysis is fantastic. It's cheap, okay, for the patient. It's a good indicator what problem might be going on. You know, sometimes doctors can't look at you and see signs and symptoms and get an honest to goodness answer. So one of the first tests they might do is a urinalysis. Well, that's pretty cheap, and it can give them an indicator with, oh, you know, all those symptoms I physically saw and measured can lead me to these three things. And now I've done a urinalysis and I've cut out two of those possible diagnoses, and I've got a more accurate diagnosis, if you will. There is something called pyelonephritis. That is true kidney infection. So not only did you have a UTI, you didn't treat the UTI, you thought that cranberry juice would fix the problem. It's not a medicine, it just aids in it. But the UTI has actually gotten worse. It has crawled up the ureters, and not necessarily crawls, but the bacteria has moved and colonized in large formations inside the kidney itself. And now it can be spread in your blood. So it's actually dangerous. And you can see that bottom image is a kidney. And you remember they look pretty nice and healthy, and this is mushy and slimy. And you can see all those holes that are not normally found in a kidney. That's from the bacteria growing or colonizing in those areas. And obviously that kidney is not functional anymore, because as I've said, kidneys are very, very particular. They're highly sensitive, so it's very easy to disrupt the kidney's function. What do? Massive amounts and very strong antibiotics are given, and if it doesn't fix, then removal of the kidney that's infected does occur. Okay. Now we're moving from the kidneys and working our way down. We have two slender tubes. Each runs behind the peritoneum, and they run from the renal hilum to the posterior backside of the bladder. Okay. These ureters are basically passageways that carry urine from the kidneys to the bladder. They don't just move. Urine doesn't just fall from the kidneys to the bladder. It doesn't move by gravity. Rather, smooth muscles contract around the ureters and propel urine into the bladder by that peristalsis, by that smooth muscle contractions. And that's actually what kidneys, ureters, and the bladder looks like. So those are straight dissections from a human cadaver. Okay, I put this here because some students definitely are affected by this, or they know somebody in their family that's affected by this. This is renal caliculi, also known as kidney stones. They're made up of tiny crystals of different types. So imagine, if you will, a crystal being formed. Well, that crystal's sticky. So if several crystals are formed by different types of ions, they make like a snowball, if you will, a ball of crystals. And it's not just smooth, it's rough and jagged. So causes of these renal caliculi or these kidney stones depends on the type of stone. It can develop over weeks or even months. So the most common one are calcium stones, which you can see in that x-ray, and they're easily identified because you can see bones in your body, and bones are made up of calcium. So if you have a calcium stone, it's very well identified on a simple x-ray. Calcium stones are most common cause, and it's because calcium combining with something in your natural biochemistry causes this snowball of calcium stones. Okay. Uric acid is another type uric acid stones are caused by gout, which you've heard about. That's where you have uric acid building up in your joints, or chemotherapy can cause it. So symptoms of having renal caliculi or kidney stones is pain, it's fever, it's chills. It's almost like a UTI on steroid, especially with the pains. Urinalysis lets us know, kidney function tests also lets us know, and x-ray lets us know. And depending on the size of it, depending on the numbers of these kidney stones, lets us decide how we're going to treat it. And there's several ways. You can pass it yourself, which from my understanding, which I've never was fortunate enough never to have to do, it can pass itself. And imagine like trying to urine out through your urethra something like shards of glass. Okay. Another thing, if there's several of them or it's too big to pass, we can try waves, if you will. Waves trying to break it down. So natural waves itself. If that doesn't occur, we tried to do surgical intervention. So at least we can break these, if the waves break it up, you're going to have to pass several of these crystals. The bladder itself is a smooth muscle capsule. It is a temporary storage facility for urine. It is just posterior to the pubic symphysis. Remember that area of the two pubic bones meeting. It has 3 openings within it. 2 of them are for ureters, 1 is for a urethra. There is a specific region that's very important to know. It is that triangular shaped one you can see on the left diagram. That's called the trigone. This is where generally infections occur. This is where a UTI would occur. Think about it. That's where the urethra is. Bacteria is introduced by the urethra. The bacteria, if you will, climbs into the urinary bladder, and that's the first area it would come in contact with. Think about it. It's warm. It has a food source. It's moist environment. That's optimal for bacterial growth. So the bladder itself has 3 layers of smooth muscle, and it is made up of mucosa, transitional epithelium. Remember transitional epithelium, you learned about it in A&P1. And some note, as the bladder amount of urine increases, it expands and rises superiorly rather than side to side. So it has to kind of raise as the volume increases of urine rather than move side to side and grow in that manner. So as urine fills, it goes this way because of its location. It does not grow this way or expand, if you will, that way. Okay. As we're moving out of the urinary bladder, we have the urethra. You have one. It's a thin-walled tube that carries urine from the bladder to outside the body. Again, not by gravity, but by peristalsis, those smooth rhythmic contractions. There are 2 sphincters involved with the urethra. Remember, we talked about sphincters before in digestive system. There's 2 sphincters. The internal urethra sphincter is made up of smooth muscle. It's involuntary. That's the one that says, oh, I think I've got to pee soon. I've got to urinate quickly. Okay. This keeps the urethra closed when urine is not being passed. So it's held in that position, and you get that sensation that you have to pee. The second sphincter is the external urethral sphincter. This is made up of skeletal muscle and is voluntary. That's when you literally go to urinate. You allow yourself to urinate. There's that UTI. We already talked about a urinary tract infection. It can occur anywhere in the urinary tract, between the urethra and the kidneys. It's usually caused 99.9% by bacteria. And you know the symptoms to it. We test it by urinalysis. You can see that sample there. That's not chicken soup. That is urine sample. And you can see it looks like chicken soup. It has that color. You can't see through it, so it's cloudy. The term for that cloudy, you can't see through it, is called turbid. This urinary sample, this urine sample is turbid. It shows turbidity. It is cloudy. Micturition is the fancy term for peeing, for urinating. Micturition. It is the act of emptying the bladder. Okay. So that two sphincter controls the flow. Stretching the bladder more than 200 milliliters activates stretch receptors. Impulses travel to the sacral region of the spinal cord and then back to the bladder, causing the bladder to release or go. So that reflex contractions causes urine to be released. So the more that is sensed as far as it filling, the more these reflex contractions occurs, causing the bladder to open and allow the urine to go out the urethra. Well, what happens as you age? Like I said, with aging, things don't function quite the same. So there's something called incontinence. That occurs when you are not able to keep urine from leaking out of your urethra. You leak out once in a while or can't hold it at all. Causes of this incontinence can be nerve and muscular problems, mental health, because remember, the mind controls urination. Childbirth can cause it. You've heard of plenty of people that are pregnant and pee on themselves when they laugh, or even it occurs afterwards. Age can cause it to occur, like I just told you. And then injury can cause it to occur. Things that help it is medicine, bladder retraining. Just like you train yourself on eating, you can train yourself on peeing, just like, an animal like a cat. And then Kegel exercises, that's where you can cause vaginal contractions. This is for women, obviously, alone. It can cause vaginal contractions, which slows the urinary process and strengthens the muscles around that sphincter. What is important about the urinary system? How does it protect you? How does it take care of you? Well, first is fluid, electrolyte, and acid balance. Okay. Blood composition depends on three major factors: diet, cellular metabolism, and how much urine is pushed out. Okay. Kidneys play a vital role in this, in these three factors, these three things to sustain survival, because it must excrete those nitrogenous wastes. It also must maintain water and electrolyte balance in the blood to ensure proper pH. So think about it. If you're urinating out lots of water, or too much water, if you're urinating out too much electrolytes, then your pH would be off. So if you are healthy, water accounts for half or more of your body weight. You can see it varies in men and women. Obviously, the amount of water it's present depending on what's going on in their life. And again, the amount of muscle they have dictates it. What's going on in their body dictates it. Water is necessary for dissolving all solutes in the body. It is considered the universal solvent. So it is a solvent. Therefore, a solvent is something that can break down a solute. Think about it. If you look at your salt, is it breaking down? Well, what happens if you put salt in water? It breaks down. You have a mixture, if you will, where you can't physically see the salt, but you know it's present if you taste it. Okay? Water is going to be found three main areas or three main locations within the body. You have intracellular fluid. Intra means inside. Intracellular fluid is water found within a cell. Extracellular fluid means outside. Extracellular fluid is body fluids outside of cells. And then lastly, where you can find water is in blood plasma. Okay? Again, remember that link between water and salt. Okay? Salt holds the central position in fluid and electrolyte balance for homeostasis, that balance. Regulating salt input and output is very important in the renal function. Water is going to follow wherever salt goes. So think about it. If we have too much water in our blood volume, then we want to push it out through the urine. So salt will go out in the urine, water will follow it. If we have not enough water inside our blood volume, then salt will not be allowed out in the urine. It will stay in blood circulation, therefore water will stay in circulation as well. Okay, because plasma is formed everywhere in the body, because blood plasma also connects internal and external environments, so exchanges between just the three systems that I have on here, respiratory system, digestive system, and kidney system, or urinary system, they all have that same blood plasma going through each of them. So think about it. If we have an issue in any of our systems, but I have these three systems present, if there is an issue of pH, if there is an issue of excess electrolytes, if I fix it, if I was a doctor and fix it in one of these systems, essentially, since plasma is found everywhere, it will fix it everywhere in the bottom. That's hypothetical. Does it always work? No, because there's secondary issues. But theoretically speaking, because blood plasma is found in each area, if we regulate a problem within the lungs, theoretically, if it is a blood plasma issues, we can regulate it throughout the body. That's the whole premise of this. So there has to be a way to regulate the water intake and outtake. Again, that has to do with hydration. It can be that thirst mechanism. That's where there is a large volume change. Roughly between 5 and 10% of plasma volume has changed. You've lost it. Okay. Normally, you know, if there's issues, that's osmolarity, we can move water where it needs to go. But when you lose that amount, that 5 to 10% of water, then you have to have an emergency measure to fix it. And there's that thirst mechanism. I mean, think about it. You've exercised a lot and you have that deep thirst where nothing, soda, juice, any of those things don't satisfy it, where you literally physically have to drink water. That is the thirst mechanism. That is your body's way to say, man, you've lost a large amount of water. We need to replace it. Okay, water is going to leave the body in a few ways. Sweating. or urine. So those are the two ways that it leaves the body. Those are the two ways that it can potentially cause this hydration, this thirst mechanism to occur. Remember, I told you that I was going to mention the hormones that are involved with urination. The hormones that are listed there affect the levels of water, of electrolytes, and ion balances, the sodium balance, if you will. So antidiuretic hormone, that's going to cause you to urine. So you can see what's going to happen with that. Aldosterone is what's going to keep the water in your bloodstream. So they're opposing aspects, so that what? Those are both opposites in a negative feedback, kind of like when we talked about sugar and insulin. Same idea. Why are electrolytes important? What exactly are electrolytes? Electrolytes are charged particles, like hydrogen. It has a positive. Sodium has a positive. Chloride has a negative. Those are all electrolytes. And we need it balanced. Electrolytes are obtained from food that is ingested, or even fluids that are ingested. Some are ingested in too much, like salts. So it's like thinking about Gatorade. Well, you just worked out, you're sweating profusely, you're at, training, football training, and you've killed it for 2, 3, 4 hours. So what is one of the things that you need to ingest? And it's Gatorade, because it's not only water in the Gatorade, but it's also electrolytes present. It's charged ions that are present that'll take care of you and keep you healthy. However, drinking nothing but Gatorade is also very dangerous. So electrolytes in high doses can irritate nerve cells. It can cause pain. It can also cause things to be stimulated that are not necessary, that action potential being moved, the contractions being moved, Okay, so that electrolyte balance is very important. I've mentioned acid-base balance as well. So all biochemical reactions are influenced by the pH of their fluid environment. Therefore, this acid-base balance needs to be closely regulated. Remember that blood pH needs to be between 7.35 and 7.45. That's on the normal side or the neutral side, with a slightly basic amount. Okay? Alkalosis is where the pH of the arterial blood rises above that 7.45. Not good. Acidosis is a drop in the arterial pH below that 7.35. So we're definitely going into that acidic range. So how do we balance it? Well, first way we balance it is, remember, we need to aid in attachment. So if we have it too acidic, remember, acids are proton donors. Just think of H+. Strong acids break apart completely in water. Remember, water is the universal solvent. So acids will break apart in water. That strongly affects the pH. It lowers or increases the pH. It goes from acidic to more neutral. Bases themselves are proton acceptors. Remember that H positive. So if it's a base, if it's above that normal neutral range, it is a proton acceptor. Right? So the idea is we want to introduce the opposite into the system to maintain or keep that neutral range. Buffers are the best way to attach to those excess hydrogen or proton, so pH won't get altered. So buffers are the best way to attach to H positive, so that that pH remains in that neutral range. Within the body, there are 3 buffer systems. There's the bicarbonate buffer system, there's the protein buffer system, and the phosphate buffer system. These are systems that fix the pH level in one of 3 fluid areas. Remember, water was found in 3 areas. It was found in blood plasma, water is found in intracellular fluid, and water is also found in interstitial fluid. Okay, so those 3 buffer systems all are involved with fixing one of those 3 areas or altering one of those 3 areas where water is stored. The only one I really want you to focus on in the most part is going to be the bicarbonate buffer system. That's where you take a strong acid, you mix it with a weak base, and what do you get? You get a weak acid that your body can handle and salt which your body can break down. That's buffering it. That's bringing it into that normal range. 2 areas control this system. First, the respiratory system controls it. Respiratory works together with the renal system, which is the second system we'll talk about, to control the pH. Remember, when you talked about respiratory system previous to this, carbon dioxide enters the blood from tissue and converts to a bicarbonate ion. So CO2 comes from the tissues, it's converted into a bicarbonate ion, which will then be transported in plasma. Now, that bicarbonate system, you already know that bicarbonate ion can go to the respiratory system where it's going to be released, but it also can be used as a buffering agent to regulate pH. Okay? Acidosis, remember, that's where the pH of blood goes below 7.35. Acidosis activates the respiratory center to increase respiratory rate, which eliminates more carbon dioxide and causes blood pH to rise. Remember, acidosis, the pH is around 2, let's say, or 3. Respiratory center increases respiratory rate, which more CO2 is pushed out. Therefore, pH will rise back to that 7.35 to 7.45 range. The second mechanism that helps control with that bicarbonate rate is going to be obviously the renal system. That's going to, the kidneys itself are going to excrete bicarbonate ions not needed, or they're in excess volumes, that therefore conserving or reabsorbing bicarbonate ions needed. Step 2, remember, bicarbonate ions then will be pulled from the kidney, from the nephron itself in step 2. So we're talking about the loop of Henle and then put back into blood volume. And the last slide that I talk about is kidney failure or renal failure. Kidneys fail to adequately filter waste products from the blood. An acute kidney injury can cause this. Generally speaking, something acute that occurs to the kidneys can be reversible with treatment. So you can have acute kidney failure due to surgery. When you're signing your life away, you know, when they make you sign all these things, these documents before you go into surgery, it's letting you know that the surgery can cause more harm than good. You theoretically, potentially could die from it. And one of the things that can occur is kidney shutdown. So a decrease in the GFR can cause, or can, I'm sorry, tell that this is a problem. Signs and symptoms can vary. Early stages can be very few and far between. Late stages can look like, kidney infection, small amounts of urine, pain definitely. What do we do? Well, you see the bypass there of blood, so it's acting, that machine there is acting like kidneys. It's not a long-term situation. And there you can see that's a kidney. Now you remember what they look like initially. That's what a kidney looks like with kidney failure. Slimy, definitely non-functioning.